Good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12th, 2011, 12:19 a.m. Eastern Time. Quick note. Various phosphate-containing compounds in which every phosphorus atom is the radioisotope phosphorus-32 are readily available. Because of their high specific activity, 32P labeled nucleotides are routinely used to label nucleic acids in cell-free systems, as discussed in the next section. However, phosphorus-32 is not suitable for certain types of incorporation experiments designed to localize labeled materials within the cell. The radioisotope iodine-125, which is also available in almost pure form and can be covalently linked to a protein or nucleic acid to yield preparations with a high specific activity. Such attachment of iodine-125 can be achieved enzymatically or chemically and generally does not drastically affect the preparations and properties of a macromolecule. End. Good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 12:22 a.m. Eastern Time. Quick note. Radio-labeled molecule can be detected by visual and quantitative methods. Depending on the nature of the experiments, labeled compounds are detected by autoradiography, semi-quantitative visual assay, or their radioactivity is determined in an appropriate counter, highly quantitative assay that permits the concentration of the radio-labeled compound to be estimated. In some experiments, both types of detection are used. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 12:24 a.m. Eastern Time. Quick note. Autoradiography. In autoradiography, a cell-to-cell constituent labeled with a radioactive compound and then overlaid with a photographic emulsion sensitive to radiation. Development of these emulsion yields smaller silver grains whose distribution corresponds to that of the radioactive material. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 12:26 a.m. Eastern Time. Quick note. Audio-radiographic studies of whole cells have been crucial in determining the intercellular site where various macromolecules are synthesized and their subsequent movements within the cells. For example, when the cells are incubated for a short time, 3H-thymidine, a unique DNA precursor, most of the radioactivity is localized to the nucleus, identifying the nucleus as a major site of DNA synthesis. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 12:28 a.m. Eastern Time. Quick note. Even after cells are incubated for prolonged periods with 3H-thymidine, virtually all of the radioactivity remains in the nucleus, indicating that the DNA remains there. Similarly, the site of synthesis of RNA is revealed by incubating cells for 1 minute with 3H-uridine, a unique RNA precursor. In this case, all the audio-radiographic grains are found over the nucleus after a longer period of incorporation. However, many audio-radiographic grains are located over the cytoplasm, indicating that RNA, in contrast to DNA, is transported from the nucleus. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 12:31 a.m. Eastern Time. Quick note. The same type of experiment with radio-labeled amino acids identifies the cytoplasm as the site of synthesis of all cellular proteins, as discussed in Chapter 16. Transparent to secretory proteins from their site of synthesis on the rough endoplasmic reticulum to the cell surface where they are secreted was first documented by the technique called electron micrograph audio-radiography, which allows each silver filament resulting from a radioactive disintegration to be observed. End. Good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 12:38 a.m. Eastern Time. Quick note. In audio-radiographic studies, the ability of the experimenter to localize the site at which the radioisotope is incorporated is affected by the energy of the particles emitted during the radioactive disintegrations. For example, the B particles emitted by phosphorus-32 are so energetic that the streaks they make on the photographic emulsion can be as long as 1 millimeter, much longer than the diameter of the individual cells. In contrast, the B particles emitted by tritium create tracks on the photographic emulsion that are only about 0.47 microns long. Thus, 3H labeled structure can be located within cells to an accuracy of about 0.5-1.0 microns, or about 1/5 the diameter of the nucleus of mammalian cells. Because tritium emits least energetic particles of all the common radioisotopes, it is highly preferred for locating labeled compounds or structures within cells. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 12:42 a.m. Eastern Time. Quick note. Quantitative determination of radio-labeled compounds. The amount of radioactivity in labeled material can be measured with either of two different instruments, a Geiger counter or scintillation counter. The former detects ions produced in gas by B particles or Y rays, gamma rays emitted from the radioisotope. In a later radio-labeled sample is mixed with a liquid containing a fluorescent compound that emits a flash of light when it absorbs the energy of the B particles of Y gamma ray released during decay of the radioisotope phototube, and the instrument detects and counts these light flashes. And good morning, this is Dr. Will, CBMD, PhD. It's Wednesday, October 12, 2011, 12:49 a.m. Eastern Time. Quick note. Unusual experimental design of radioactive precursor is incubated with whole cells or cell-free extracts. Cellular constituents and are isolated and purified in various ways. Finally, the radioactivity of the various fractions is measured with a counter. For example, to identify the site of RNA synthesis, cells can be incubated for a short period with 3H-uridine and then subjected to fractionation procedure to separate the various organelles. The specific activity of the nucleus fraction dPm divided by mg protein is found to be much higher than that of any other organelle fraction, thus confirming the nucleus as the site of RNA synthesis. And good morning, this is Dr. Will, CBMD, PhD. It's Wednesday, October 12, 2011, 12:52 a.m. Eastern Time. Quick note. A combination of labeling and biochemical techniques and of visual and quantitative detection methods are often employed. For instance, to identify the major proteins synthesized by a particular type cell, a sample of the cells is incubated within a radioactive amino acid, 35S labeled methionine, for a few minutes. The mixture of cellular proteins then is resolved by gel electrophoresis, and the gel is subjected to radioactive bands corresponding to newly synthesized proteins which have incorporated a radio-labeled amino acid. Alternatively, the proteins can be resolved by liquid chromatography and of radioactivity in the eluted fractions determined quantitatively with a counter. And good morning, this is Dr. Will, CBMD, PhD. It's Wednesday, October 12, 2011, 1:01 a.m. Eastern Time. Quick note. Intracellular precursor pools affects the outcome of pulse chase experiments. The purpose of some studies is to label macromolecule and then trace the labeled compound as it moves through various cellular compartments or is incorporated into other molecules within cells. This can be accomplished with a pulse chase experiment in which cells are incubated briefly with a radio-labeled precursor, the pulse followed by removal and replacement of a labeled precursor by an excess of unlabeled precursor. The chase, the fate of the newly synthesized compound that incorporated the label during the pulse, is tracked by measuring the radioactivity of the cells or cell fractions at various times thereafter. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 1:29 a.m. Eastern Time. Quick note. In this type of experiment, the small labeled precursor added to the medium, an amino acid nucleoside or a phosphate ion, first enters an intercellular pool of small molecules which are free to diffuse through the cytoplasm and nucleus. Subsequently, the precursor is incorporated into the macromolecule, protein, or nucleic acid, depending on the growth conditions of a cell culture. The quantities of compounds in the intercellular pool may vary. Likewise, the rates at which different precursors are absorbed, utilized, and secreted by cell varies. Both of these characteristics of small molecules and the soluble pool must be considered in the design and interpretation of pulse chase experiments. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 1:31 a.m. Eastern Time. Quick note. The clearest pulse chase effect occurs when the labeled precursor is rapidly exchanged between the intercellular pool and external medium. In this case, the labeled precursor that accumulates the cells during the pulse is rapidly depleted during the chase, thus incorporated the label into a macromolecule soon ceases, and the radioactivity is associated only with macromolecules synthesized during the pulse. As shown, pulse chase experiments with labeled amino acids exhibit this pattern because rapid equilibrium occurs between amino acids into and inside the cells in the medium. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 1:35 a.m. Eastern Time. Quick note. Ribonucleosides and deoxyribonucleosides, however, become phosphorylated soon after they enter the cell pool, and phosphorylated compounds do not generally leave the cell. Thus, labeled nucleosides can enter the cell, but no equilibrium is established between the nucleic acid precursors in the medium and their phosphorylated counterparts in the cell. Nevertheless, a practically useful pulse chase effect can be obtained in experiments with radioactive deoxyribonucleosides because the deoxyribonucleotide contends to the cell pool is sufficient for only a few minutes of DNA synthesis. Labeled thymidine, for example, can be satisfactorily chased even though it is phosphorylated to TMP and TTP because an amount of TTP equal to that in the pool is utilized every few minutes for replication of DNA. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 3:30 a.m. Eastern Time. Quick note. As members of the same family containing the same functional group, alkyl halides of different classes tend to undergo the same kinds of reactions. They differ in rates of reaction, however, and these differences in rates may lead to other deeper differences. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 3:32 a.m. Eastern Time. Quick note. We should notice that similar names do not always mean the same classification. For example, isopropyl chloride is a secondary chloride, whereas isobutyl chloride is a primary chloride. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 3:38 a.m. Eastern Time. Quick note. To begin with, alcohols are the compounds from which alkyl halides, in fact most substances undergoing nucleophilic substitution, are nearly always made. Next, once we have made alkyl halides and are allowing them to undergo nucleophilic substitution, we encounter alcohols again playing a variety of roles. They may be the reagents with which alkyl halides react, or the products or the solvents in which reaction takes place. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 3:39 a.m. Eastern Time. Quick note. Alcohols are compounds of the general formula ROH, where R is an alkyl group and OH is a hydroxyl group. A simple alcohol can be named just by naming the alkyl group that holds the hydroxyl group and following this with the word alcohol. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 3:40 a.m. Eastern Time. Quick note. Like an alkyl halide, an alcohol is classified as primary, secondary, or tertiary according to the kind of carbon that bears the OH group. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 4:06 a.m. Eastern Time. Quick note. Of the varied chemical properties of alcohols, there is just one pair that we should become acquainted with at this point: their acidity and basicity. These properties reside in the hydroxyl group, OH functional group of alcohols. This group is like the hydroxyl group of water, a compound with which we are already familiar. Like water, alcohols are weak acids and weak bases, roughly about as acidic and about as basic as water. Like water, alcohols are acidic enough to react with active metals to liberate hydrogen gas. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 4:09 a.m. Eastern Time. Quick note. The products are called alk oxide, sodium eth oxide, for example, or potassium and peroxide. Alk oxides are strong bases like sodium hydroxide. Like water, alcohols are basic enough to accept a proton from strong acids, like hydrogen chloride and hydrogen sulfate, and thus bring about complete dissociation of these compounds. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 4:50 a.m. Eastern Time. Quick note. Early Celtic Stonehenge discovered in Germany's Black Forest. Science Daily, October 11, 2011. A huge early Celtic calendar construction has been discovered in the royal tomb of Magdalenberg. This discovery was made by researchers in Germany when they evaluated old excavation plans. The order of the burials around the central royal tomb fits exactly with the sky constellations of the northern hemisphere. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 4:53 a.m. Eastern Time. Quick note. Whereas Stonehenge was oriented toward the sun, the more than 100-meter width burial mound of Magdalenberg was focused towards the north. The builders positioned long rows of wooden posts in the burial mound to be able to focus on the lunar standstills. These lunar standstills happen every 18 to 6 years and were the cornerstones of the Celtic calendar. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 4:55 a.m. Eastern Time. Quick note. The position of the burials at Magdalenberg represents a constellation pattern which can be seen between midwinter and midsummer. With the help of special computer programs, researchers could reconstruct the position of the sky constellations in the early Celtic period and following from those which were visible in midsummer. These archaeoastronomical research resulted in the date midsummer 618 BC, which makes it the earliest and most complete example of a Celtic calendar focused on the moon. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 4:57 a.m. Eastern Time. Quick note. These types of calendars were completely forgotten in Europe, but the Romans, a sun-based calendar, was adopted throughout Europe. The full dimensions of the lost Celtic calendar system have now come to light, again in the monumental burial mound of Magdalenberg. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 5:16 a.m. Eastern Time. Quick note. Why climate models underestimate Arctic sea ice retreat. No Arctic sea ice in summer by end of the century. Science Daily, October 12, 2011. In recent decades, Arctic sea ice has suffered a dramatic decline that exceeds climate model predictions. The unexpected rate of ice shrinkage has now been explained by researchers at CNRS. They argue that climate models underestimate the rate of ice thinning, which is actually about 4 times faster than calculations. This model bias is due to the poor representation of the sea ice southward drift out of the Arctic basin through the Frame Strait, when this mechanism was taken into account to correct the discrepancy between simulations and observations. Result from the new model suggested that there will be no Arctic sea ice in the summer by the end of the century. And good morning, this is Dr. Will, CBMD, PhD. It is Wednesday, October 12, 2011, 5:18 a.m. Eastern Time. Quick note. The Arctic has been losing about 10% of its permanent ice layer every 10 years since 1980. Melting of Arctic sea ice has also reached record heights in mid-September 2007. At the point when sea ice reaches its annual minimum extent, perennial ice covered an area of 4.14 million kilometers to the power of 2. This record low level was nearly reached again in September 2011. 4.34 million kilometers to the power of 2 climate simulations conducted for the IPCC-2 simulate the decline of Arctic ice resulting from global warming. They predict that summer ice will disappear altogether at the end of the century. And good morning, this is Dr. Will, CBMD, PhD. It's Wednesday, October 12, 2011, 5:20 a.m. Eastern Time. Quick note. However, when compared with 30 years of detailed satellite observations, these models appear optimistic. Arctic sea ice has thinned on average 4 times faster over the period of 1979 to 2008 than in climate simulations. True observations are therefore not correctly reproduced by these climate models, which were mainly calibrated using global variables, such as world average rather than regional temperature. And good morning, this is Dr. Will, CBMD, PhD. It is Wednesday, October 12, 2011, 5:27 a.m. Eastern Time. Quick note. The drift of sea ice is poorly described by the models, which do not take drift acceleration or southward evacuation of the sea ice into account. Modeled sea ice behaves as though it drifts freely without any mechanical interaction between ice fragments. Whatever the season, period, or ice thickness, there is no link in the models between the thinning of the ice and the further acceleration of its drift. And good morning, this is Dr. Will, CBMD, PhD. It is Wednesday, October 12, 2011, 5:29 a.m. Eastern Time. Quick note. This mechanism suggests that well before the end of the century, the Arctic Ocean will be devoid of sea ice in late summer. The disappearance of Arctic sea ice will probably occur in the next few decades, with far-reaching consequences for ecosystems, sea routes, and offshore exploitation of resources. And good morning, this is Dr. Will, CBMD, PhD. It is Wednesday, October 12, 2011, 5:40 a.m. Eastern Time. Quick note. Liquid can turn into solid under high electric field, physicists show in simulations. Science Daily, October 10, 2011. Physicists have demonstrated in simulations that under influence of sufficiently high electric fields, liquid droplets of certain materials will undergo solidification, forming crystallites at temperature and pressure conditions that correspond to liquid droplets at field-free conditions. This electric field-induced phase transformation is termed electrocrystallization. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 5:41 a.m. Eastern Time. Quick note. The study performed by scientists at the Georgia Institute, the technology appears online, is scheduled as a feature and cover article in 42nd issue of volume 115 of the Journal of Physical Chemistry C. And good morning, this is Dr. Will, CBMD, PhD. It is Wednesday, October 12, 2011, 5:42 a.m. Eastern Time. Quick note. While liquid drops under field-free conditions are spherical, they alter their shape in response to an applied electric field to become needle-like liquid drops. And good morning, this is Dr. Will, CBMD, PhD. It is Wednesday, October 12, 2011, 5:45 a.m. Eastern Time. Quick note. With the use of molecular dynamics simulations developed at the CCMS, which allow scientists to track the evolution of material systems with ultra-high resolution in space and time, the physicists explored the response of the formaldehyde nanodroplet to an applied electric field of variable strength, influenced by the field of less than 0.5 V divided by Nm. The spherical droplet elongated only slightly. However, when the strength of the field was raised to a critical value close to 0.5 V divided by Nm, the simulated droplet was found to undergo a shape transition resulting in a needle-like liquid droplet, with its long axis orientated along the direction of the applied field, measuring about 12 times larger than the perpendicular cross-sectional small axis of the needle-like droplet. The value of the critical field found in the simulations agrees well with a prediction obtained almost half a decade ago by Taylor from general macroscopic considerations. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 5:47 a.m. Eastern Time. Quick note. Past the shape transition further increased, the applied electric field yielded a slow, gradual increase of the aspect ratio between the long and short axis of the needle-like droplet, with the formaldehyde molecules exhibiting liquid diffusional motions. And good morning, this is Dr. Will, CBMD, PhD. It's Wednesday, October 12, 2011, 5:49 a.m. Eastern Time. Quick note. Here came the eureka moment, when the field strength in the simulation was ramped up even further, reaching a value close to 1.5 V divided by Nm. The liquid needle underwent a solidification phase transition exhibiting by a freezing of a diffusional motion, culminating in the formation of a formaldehyde single crystal characterized by a structure that differs from that of the X-ray crystallographic one determined years ago under the zero field conditions. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 5:55 a.m. Eastern Time. Quick note. Further analysis has shown that crystallization transition involved arrangement of the molecules into a particular spatial ordered lattice, which optimizes the interactions between the positive and negative ends of the dipoles of neighboring molecules, resulting in minimization of the free energy of the resulting rigid crystalline needle. When the electric field applied to the droplet was subsequently decreased, the crystalline needle remelted, and at a zero field, the liquid droplet reverted to a spherical shape. The field reversal process was found to exhibit a hysteresis. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 5:57 a.m. Eastern Time. Quick note. This investigation unveiled fascinating properties of a large group of materials under the influence of applied fields. Here the field-induced shape and crystallization transition occurred because formaldehyde, like water and many other materials, is characterized by a relatively large electric dipole moment. The study demonstrated the ability to employ external fields to direct and control the shape, the aggregation phase, that is, solid or liquid, and the properties of certain materials. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 6:01 a.m. Eastern Time. Quick note. Along with a fundamental interest in understanding the microscopic origins of material behavior, this may lead to development of applications of field-induced materials control in diverse areas ranging from targeted drug delivery, nanoencapsulation, printing and nanostructures, and surface patterning, to aerosol science, electrospray propulsion, and environmental science. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 6:21 a.m. Eastern Time. Quick note. Answer of the day. What is a gyroscope used for? A gyroscope, a kind of spinning top that is held in two or more circular frames, each orientated along a different axis, is used in compasses and automatic pilots on aircraft and ships. In the steering mechanism of torpedoes and anti-roll equipment on large ships. In the mid-19th century, Jean Bernard Leon Foucault invented the Foucault pendulum, which helped him explain that the Earth rotates on its axis. The work also led to his development of the gyroscope. Foucault took the name from the Greek words gyros and scopion, meaning rotation and to view. Some 50 years later, American scientist Elmer Ambrose Sperry perfected the use of gyroscopes. In 1909, he developed the first automatic pilot, which uses a gyroscope sense of direction to maintain the course of an airplane. His gyro compass was installed on the U.S. battleship Delaware in 1911. Elmer Sperry was born on October 12, 1860. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 8:10 a.m. Eastern Time. Quick note. Worms among first animals to surface after KT extinction event. Study finds. Science Daily, October 10, 2011. A new study of sediments laid down shortly after an asteroid plowed into the Gulf of Mexico 65.5 million years ago. An event that is linked to widespread global extinctions, including the demise of big dinosaurs, suggests that lowly worms may have been the first fauna to show themselves following the global catastrophe. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 12, 2011, 8:12 a.m. Eastern Time. Quick note. While the focus on the so-called KT boundary extinction is often on the survival and proliferation of mammals, paleobotanical studies show some of the earliest terrestrial ecosystems to emerge were dominated by low diversity and opportunistic aquatic plants. University of Colorado Boulder Geological Science Associate Professor. While sediments down immediately following the impact event generally have relatively few animal fossils, new evidence from North Dakota shows networks of crisscrossing burrows less than 3 inches above the KT boundary layer. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 12, 2011, 8:15 a.m. Eastern Time. Quick note. Fossil burrows provide direct evidence of animal activity that occurred right at the spot. These burrows are quite extensive. Their characteristics suggest they were probably produced by worms. To my knowledge, such burrows haven't been documented in terrestrial environments this close to the KT boundary. This is a glimpse of the world we don't know very much about yet. And Good morning, this is Dr. Will, CBMD, PhD. This Thursday, October 13, 2011, 12:26 a.m. Eastern Time. Quick note. Eating your greens can change the effect of your genes on heart disease. Science Daily, October 12, 2011. A long-held mantra suggests that you can't change your family, the genes they pass on, or the effect these genes. Now, an international team of scientists led by researchers at McMaster and McGill universities is attacking that belief. And good morning, this is Dr. Will, CBMD, PhD. This Thursday, October 13, 2011, 12:27 a.m. Eastern Time. Quick note. The researchers discovered the gene that is the strongest marker for heart disease can actually be modified by generous amounts of fruit and raw vegetables. The results of their study are published in the current issue of the journal PLOS Medicine. And good morning, this is Dr. Will, CBMD, PhD. This Thursday, October 13, 2011, 12:30 a.m. Eastern Time. Quick note. We observe that the effect of high risk of genotype can be mitigated by consuming a high diet in fruits and vegetables. Results support the public health recommendation to consume more than 5 servings of fruits or vegetables as a way to promote good health. And good morning, this is Dr. Will, CBMD, PhD. This Thursday, October 13, 2011, 12:33 a.m. Eastern Time. Quick note. Astrophysicists find evidence of black holes' destruction of stars. Science Daily, October 12, 2011. Astrophysicists have found evidence of black holes destroying stars, a long-sought phenomenon that provides a new window into general relativity. The research reported in the latest issue of the Astrophysical Journal also opens up a method to search for the possible existence of a large population of presently undetectable intermediate mass black holes, which are hypothesized to be precursors to the supermassive black holes at the centers of most large galaxies. And good morning, this is Dr. Will, CBMD, PhD. This Thursday, October 13, 2011, 12:36 a.m. Eastern Time. Quick note. Cosmologists have calculated that on occasion the star's orbit will be distributed in such a way that it passes very near the supermassive black hole at the center of its galaxy, but not so close that it is captured whole. Such a star will be torn apart by the extreme tidal forces it experiences. The force of gravity on the near side of the star is so much stronger than that on the far side that its gravitational force holding the star together is overwhelmed, causing the star to simply come apart. While some of the star's matter falls into the black hole, much of it continues in chaotic orbits, crashing into itself and producing intense radiation lasting days to months. These phenomena are called stellar tidal disruption flares, or TDFs. And good morning, this is Dr. Will, CBMD, PhD. This Thursday, October 13, 2011, 12:37 a.m. Eastern Time. Quick note. Although discovering evidence of TDFs has been a high priority of astrophysicists for many years, and several possible examples have been found using X-ray and UV satellites discovering TDFs in large-scale systematic survey using ground-based optical telescopes, as has now been achieved, is critical to controlling bias and avoiding misidentifications. And good morning, this is Dr. Will, CBMD, PhD. This Thursday, October 13, 2011, 6:58 a.m. Eastern Time. Quick note. The difficulty in detecting TDFs is largely due to the challenge of distinguishing them from more common types of flares such as supernova. For every TDF there are about 1,000 supernovae. In addition, some supermassive black holes have an accretion disk surrounding them. Gas and dust often left from an earlier merger with another galaxy, which is continuously feeding the hole. Such accreting black holes are usually evident from the bright emission they produce and are known as quasars, or active galactic nuclei, AGN. However, a hiccup in the accretion of an undetected active black hole could produce a flare that might be mistakenly identified as a TDF. And good morning, this is Dr. Will, CBMD, PhD. This Thursday, October 13, 2011, 6:59 a.m. Eastern Time. Quick note. These almost all could be classified into supernova and AGN flares. However, 2 cases were left that did not fit either classification by relying on multi-year observations. The researchers could see that the 2 flares' host galaxies showed no other flaring activity, as would be the case if the flares came from a hidden variable AGN. This means the possibility these 2 flares were produced by undetected AGNs is extremely small. And good morning, this is Dr. Will, CBMD, PhD. This Thursday, October 13, 2011, 7:04 a.m. Eastern Time. Quick note. Researchers reconstruct genome of Black Death, bacteria found to be ancestor of all modern plaques. Science Daily, October 12, 2011. An international team led by the McMaster University of Germany has sequenced the entire genome of the Black Death, one of the most devastating epidemics in human history. And good morning, this is Dr. Will, CBMD, PhD. This Thursday, October 13, 2011, 7:05 a.m. Eastern Time. Quick note. This marks the first time scientists have been able to draft a reconstructed genome of any ancient pathogen, which will allow researchers to track changes in the pathogen's evolution and virulence over time. This work, currently published online in the Journal of Nature, could lead to a better understanding of modern infectious diseases. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 13, 2011, 7 to 9 a.m. Eastern Time. Quick note. In a separate study published recently, the team described a novel methodological approach to pull out tiny degraded DNA fragments of the causative agent of the Black Death, and showed that a specific variant of the Yersinia pestis bacterium was responsible for the plague that killed 50 million Europeans between 1347 and 1351. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 13, 2011, 7:10 a.m. Eastern Time. Quick note. The genomic data show that this bacteria strain, the variant, is an ancestor of all modern plaques we have today worldwide. Every outbreak across the globe today stems from a descendant of the medieval plague. With a better understanding of the evolution of this deadly pathogen, we are entering a new era of research into the infectious disease. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 13, 2011, 7:11 a.m. Eastern Time. Quick note. The direct descendants of the same bubonic plague continue to exist today, killing some 2,000 people each year. We found that in 660 years of evolution as human pathogen, there have been relatively few changes in the genome of the ancient organism, but those changes, however small, may or may not account for the noted increased virulence of the bug that ravaged Europe. The next step is to determine why this was so deadly. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 13, 2011, 7:12 a.m. Eastern Time. Quick note. Major technical advances in DNA recovery and sequencing have dramatically expanded the scope of genetic analysis of ancient specimens, operating new horizons in the understanding of emerging and re-emerging infections. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 13, 2011, 7:14 a.m. Eastern Time. Quick note. Possible trigger for volcanic super eruptions discovered in Science Daily, October 12, 2011. The super eruption of the major volcanic system occurs about every 100,000 years and is considered one of the most catastrophic natural events on Earth, yet scientists have long been unsure about what triggers these violent explosions. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 13, 2011, 7:15 a.m. Eastern Time. Quick note. However, a new model presented this week by researchers at Oregon State University points to a combination of temperature influence and the geometrical configuration of the magma chamber as a potential cause for these super eruptions. Result of the research was funded by the National Science Foundation, were presented at the annual meeting of the Geological Society of America in Minneapolis, Min. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 13, 2011, 7:17 a.m. Eastern Time. Quick note. Previous modeling efforts have focused on an eruption trigger from within the magma chamber, which scientists thought would leave a visible trace in the form of a precursor eruption deposits, according to the geologists and co-author of the study. Yet there has been a distinct lack of physical evidence for precursor eruption at the site of these supervolcanoes. The model suggests the reason there may be no precursor eruption is that the trigger comes from above, not from within. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 13, 2011, 7:21 a.m. Eastern Time. Quick note. Dark matter of the genome revealed through analysis of 29 mammals. Science Daily, October 12, 2011. An international team of researchers has discovered the vast majority of the so-called dark matter in the human genome by means of a sweeping comparison of 29 mammalian genomes. The team, led by scientists from the Broad Institute, has pinpointed the parts of the human genome that control when and where genes are turned on. This map is a critical step in interpreting the thousands of genetic changes that have been linked to human disease. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 13, 2011, 7:35 a.m. Eastern Time. Quick note. Their findings appear online October 12, 2011. The Journal of Nature early comparison studies of the human and mouse genomes led to the surprising discovery that the regulatory information that controls gene dwarfs the information in the genes themselves, but these studies were indirect. They could infer existence of these regulatory sequences, but could find only a small fraction of them. These mysterious sequences have been referred to as the dark matter of the genome, analogous to the unseen matter and energy that make up most of the universe. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 13, 2011, 7:40 a.m. Eastern Time. Quick note. The scientists were able to suggest possible functions for more than half of 360 million DNA letters contained in the conserved elements, revealing the hidden meaning behind the AS, CS, TS, and GS. These reveal almost 4,000 previously undetected exons, or segments of DNA that code for protein, 10,000 highly conserved elements that may be involved in how proteins are made, more than 1,000 new families of RNA secondary structures with diverse roles in gene regulation, 2.7 million predicted targets of transcription factors, proteins that control gene expression, and... Good morning, this is Dr. Will, CBMD, PhD. This Thursday, October 13, 2011, 7:46 a.m. Eastern Time. Quick note, how life might have survived Snowball Earth. Science Daily, October 10, 2011. Global glaciation likely put a chill on life on Earth hundreds of millions of years ago, but new research indicates that simple life in the form of photosynthetic algae could have survived in a narrow body of water with characteristics similar to today's Red Sea. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 13, 2011, 7:46 a.m. Eastern Time. Quick note, under those frigid conditions, there are not a lot of places where you would expect liquid water and light to occur in the same area, and you need both of those things for photosynthetic algae to survive. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 13, 2011, 7:48 a.m. Eastern Time. Quick note, a long, narrow body of water, such as the Red Sea, about 6.5 times longer than it is wide, would create enough physical resistance to advancing glacial ice that the ice sheet likely could not make it all the way to the end of the sea before conditions cause the ice to turn to vapor. That would leave a small expanse of open water where the algae could survive. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 13, 2011, 7:49 a.m. Eastern Time. Quick note, many scientists believe Earth became a giant snowball 2 or 3 times between 800 million and 550 million years ago, with each episode lasting about 10 million years. These all preceded the Cambrian explosion about 530 million years ago, when life on Earth rapidly expanded, diversified, and became more complex. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 13, 2011, 7:52 a.m. Eastern Time. Quick note, Snowball Earth event, the open water in such a sea, wouldn't have lasted long if it didn't have a way of being replenished. If, for example, the glacial ice acted as a dam and cut off the influx of additional seawater, the open water had to exist on the order of 10 million years for the algae to survive. Over 10 million years, you could evaporate the deepest lake in the world. If you are in a desert, you'd have to have a supply of seawater. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 13, 2011, 7:53 a.m. Eastern Time. Quick note, what in mild researchers take the temperature of Mars past? Science Daily, October 12, 2011. Researchers at the California Institute of Technology, Caltech, have directly determined the surface temperature of early Mars for the first time, providing evidence that's consistent with the warmer and wetter Martian past. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 13, 2011, 7:54 a.m. Eastern Time. Quick note, by analyzing carbonate minerals in a 4 billion-year-old meteorite that originated near the surface of Mars, the scientists determined that the minerals formed at about 18 degrees Celsius, 64 degrees Fahrenheit. The thing that is really cool is that 18 degrees is not particularly cold nor particularly hot. And good evening, this is Dr. Will, CBMD, PhD. This Thursday, October 13, 2011, 11:53 p.m. Eastern Time. Quick note, erasing history temporal cloaks adjust light's throttle to hide an event in time. Science Daily, October 13, 2011. Researchers from Cornell, NY have demonstrated for the first time that it is possible to cloak a singular event in time, creating what has been described as a history editor. In a feat of Einstein-inspired physics, and his colleagues sent a beam of light traveling down an optical fiber and through a pair of so-called time lenses. Between these two lenses, the researchers were able to briefly create a small bubble, or a gap in the flow of light. During that fleeting brief moment, lasting only the tiniest fraction of a second, the gap functioned like a temporal hole, concealing the fact that a brief burst of light ever occurred. And good evening, this is Dr. Will, CBMD, PhD. This Thursday, October 13, 2011, 11:55 p.m. Eastern Time. Quick note, their ingenious system, which is the first physical demonstration of a phenomenon originally described theoretically a year ago by Martin McCall and his colleagues at the Imperial College London in the Journal of Optics, relies on the ability to use short, intense pulses of light to alter the speed of light as it travels through optical materials, in this case an optical fiber. In a vacuum, light maintains its predetermined speed limit of 180,000 miles per second as the beam passes through a split time lens, a silicone device originally designed to speed up data transfer. It accelerates near the center and slows down along the edges, causing it to balloon out toward the edges, leaving a dead zone around which the light waves curve. A similar lens a little farther along the path produces the exact but opposite velocity adjustments, resetting the speeds and reproducing the original shape and appearance of the light rays. And Good evening, this is Dr. Will, CBMD, PhD. It's Thursday, October 13, 2011, 11:57 p.m. Eastern Time. Quick note. To test the performance of their temporal cloak, the researchers created a pulse of light directly between the two lenses. The pulse is repeated like clockwork at a rate of 41 kilohertz. When the cloak was off, the researchers were able to detect a steady beat by switching on the temporal cloak, which was synchronized with the light pulses. All signs that these events ever took place were erased from the data stream. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 14, 2011, 12:02 a.m. Eastern Time. Quick note. Unlike spatial optical cloaking, which typically requires the use of metamaterials, specially created materials engineered to have specific optical properties, the temporal cloak designed by the researchers relies more on the fundamental properties of light and how it behaves under highly constrained space and time conditions. The area affected by the temporal cloak is a mere 6 millimeters long and can last only 20 trillionths of a second. The length of the cloaked area and the length of time it is able to function are tightly constrained, primarily by the extreme velocity of light. Cloaking for a longer duration would create turbulence in the system, essentially pulling back the curtain and hinting that an event had occurred. Also, to achieve any measurable macroscopic effects, an experiment of planetary and even interplanetary scales would be necessary. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 14, 2011, 12:10 a.m. Eastern Time. Quick note. T-Rex was bigger and grew faster than previously thought. Computational analyst reveals. Science Daily, October 13, 2011. A new study reveals that T-Rex grew more quickly and reached significantly greater masses than previously estimated. In a departure from earlier methods, the new study uses mounted skeletons to generate body mass estimates. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 14, 2011, 12:11 a.m. Eastern Time. Quick note. In a departure from earlier methods, the new study uses mounted skeletons to generate body mass estimates. Previous methods for calculating mass relied on scale models, which can magnify even minor errors or in extrapolations from living animals with very different body plans from dinosaurs. We overcame such problems by using the actual skeletons as a starting point for our study. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 14, 2011, 12:15 a.m. Eastern Time. Quick note. The team used 3D laser scans of mounted skeletons as a template for generating fleshed-out digital models whose masses could be then computed. The laser scans are accurate to less than a half an inch for skeletons that are up to 40 feet long. Digital body cross-sections were reconstructed along the length of each skeleton using the relationships of soft tissues of skeletons in birds and crocodiles as a guide. A digital skin was then overlaid to generate a body volume, whose mass was calculated after empty spaces such as lungs and the mouth cavity were modeled and subtracted. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 14, 2011, 12:17 a.m. Eastern Time. Quick note. In order to appreciate the uncertainty involved in estimating how much flesh would wrap the skeletons of an extinct animal, body sections, head, neck, torso, legs, tail, were modeled individually at 3 levels of fleshiness. The 3 versions of each body segment were combined in different ways to generate a range of whole body models with varying masses. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 14, 2011, 12:27 a.m. Eastern Time. Quick note. Bacterial communication could affect Earth's climate, researchers discover. Science Daily, October 13, 2011. Woods Hole Oceanographic Institution, WHOL, scientists have discovered that bacterial communication could have a significant impact on the planet's climate. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 14, 2011, 12:43 a.m. Eastern Time. Quick note. The source of carbon in the particles in atmospheric carbon dioxide, a heat-trapping greenhouse gas. Bacterial communication could lead to the release of carbon from the particles at shallower depths, rather than sinking to the ocean depths. According to the scientists, this means that bacterial communication results in less carbon dioxide being drawn out of the air and transferred to the bottom of the ocean, from where it cannot easily return to the atmosphere. This represents the first evidence that bacterial communication plays a crucial role in Earth's carbon cycle. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 14, 2011, 12:49 a.m. Eastern Time. Quick note. Researchers correct sickle cell disease in adult mice. Protein could be a target for treating people who have a blood disorder. Science Daily, October 14, 2011. National Institutes of Health funded scientists have corrected sickle cell disease in adult laboratory mice by activating production of special blood component normally produced before but not after birth. And good morning, this is Dr. Will, CBMD, PhD. It's Friday, October 14, 2011, 12:51 a.m. Eastern Time. Quick note. Sickle cell disease results from the abnormality in hemoglobin, the protein found in red blood cells that is responsible for transporting oxygen throughout the body. People living with sickle cell disease have two copies of an altered gene that produces sickle hemoglobin instead of normal adult hemoglobin. Sickle hemoglobin changes shape after releasing its oxygen, causing the red blood cell to become stiff, misshapen and sticky, and slowing blood flow to tissues. This process damages organs and causes pain. And good morning, this is Dr. Will, CBMD, PhD. It is Friday, October 14, 2011, 12:53 a.m. Eastern Time. Quick note. The study tested a new approach to increasing the production of a third form of hemoglobin. Fetal hemoglobin production of fetal hemoglobin predominates before birth, but turns off thereafter as adult hemoglobin production takes over. People with sickle cell disease are unable to make normal adult hemoglobin and instead make sickle hemoglobin starting in infancy. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 14, 2011, 12:57 a.m. Eastern Time. Quick note. The current paper details how the research team silenced the mouse gene that produces the BCL11A protein in mice with sickle cell disease. Silencing the gene turned off production of the BCL11A protein and allowed the adult mice to continue to produce fetal hemoglobin. It appears to have eliminated disease symptoms without affecting other aspects of blood production. And good morning, this is Dr. Will, CBMD, PhD. This is Friday, October 14, 2011, 7:23 a.m. Eastern Time. Quick note. Oldest fossil rodents in South America discovered. Find is 10 million years older and confirms animals from Africa. Science Daily, October 11, 2011. On a literal walk through time along the Ucayali River near Contamina, Peru, a team of researchers found rodent fossils at least 41 million years older, by far the oldest in South American continent. And good morning, this is Dr. Will, CBMD, PhD. It's Friday, October 14, 2011, 11:36 p.m. Eastern Time. Quick note. From blue whales to earthworms, a common mechanism gives shape to living beings. Science Daily, October 14, 2011. Why don't our arms grow from the middle of our bodies? The question isn't as trivial as it appears. Vertebrate limbs, ribs, tailbone. In only 2 days, all these elements take their place in the embryo in the right spots and with the precision of Swiss watch. Intrigued by the extraordinary reliability of this mechanism, biologists have long wondered how it works. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 14, 2011, 11:46 p.m. Eastern Time. Quick note. From blue whales to earthworms, a common mechanism gives shape to living beings. Science Daily, October 14, 2011. Why don't our arms grow from the middle of our bodies? The question isn't as trivial as it appears. Vertebrate limbs, ribs, tailbone. In only 2 days, all these elements take their place in the embryo, in the right spot, and with the precision of a Swiss watch. Intrigued by the extraordinary reliability of this mechanism, biologists have long wondered how it works. And good morning, this is Dr. Will, CBMD, PhD. This is Monday, October 17, 2011, 1:42 a.m. Eastern Time. Quick note. Carbon nanotube muscles generate giant twists for novel motors. Science Daily, October 14, 2011. New artificial muscles that twist like the trunk of an elephant but provide 1,000 times higher rotation per length have been developed by a team of researchers from the University of Texas, Dallas. And good morning, this is Dr. Will, CBMD, PhD. It's Monday, October 17, 2011, 1:46 a.m. Eastern Time. Quick note. Uncharted territory, scientists sequenced the first carbohydrate biopolymer. Science Daily, October 15, 2011. DNA and protein sequencing have forever transformed science, medicine, and society. Understanding the structure of these complex biomolecules has revolutionized drug development, medical diagnostics, forensic science, and our understanding of evolution and the development. But one major molecule in the biological triumvirate has remained largely uncharted: carbohydrate biopolymers. And good morning, this is Dr. Will, CBMD, PhD. It's Monday, October 17, 2011, 1:48 a.m. Eastern Time. Quick note. Subtly shaded map of moon reveals titanium treasure troves. Science Daily, October 15, 2011. A map of the moon combining observations in visible and ultraviolet wavelengths shows a treasure trove of areas rich in titanium ores. Not only is titanium a valuable element, it is key to helping scientists unravel the mysteries of the moon interior. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 19, 2011, 2:16 a.m. Eastern Time. Quick note. Sea levels to continue to rise for 500 years. Long-term climate calculations suggest so. Science Daily, October 17, 2011. Rising sea levels in the incoming centuries is perhaps one of the most catastrophic consequences of rising temperatures. Massive economic costs, social consequences, and forced migrations could result from global warming. But how frightening of times are we facing? Researchers from the Niels Bohr Institute are part of a team that has calculated the long-term outlook for rising sea levels in relation to the emissions of greenhouse gases and pollution of the atmosphere using climate models. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 19, 2011, 2:19 a.m. Eastern Time. Quick note. Based on the current situation, we have projected changes in sea level 500 years into the future. We are not looking at what is happening with the climate, but are focusing exclusively on sea levels. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 19, 2011, 2:21 a.m. Eastern Time. Quick note. He has developed a model in collaboration with researchers from England and China that is based on what happens with the emission of greenhouse gas and aerosols and the pollution of the atmosphere. Their model has been adjusted backwards to the actual measurements and was then used to predict the outlook for rising sea levels. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 19, 2011, 2:22 a.m. Eastern Time. Quick note. The research group has made calculations for 4 scenarios, a pessimistic one, an optimistic one, and 2 more realistic ones. In the pessimistic scenario, emissions continue to increase. This means that sea levels will rise 1.1 meter by the year 2100 and will rise 5.5 meters by the year 2500. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 19, 2011, 2:38 a.m. Eastern Time. Quick note. Archaeologists find blade production lines existed as much as 400,000 years ago. Science Daily, October 17, 2011. Archaeology has long associated advanced blade production with the Upper Paleolithic period, about 30,000 to 40,000 years ago, linked with the emergence of Homo sapiens and cultural features such as cave art. Now researchers at university have uncovered evidence which shows that modern blade production was also an element of industry during the late Lower Paleolithic period, 200,000 to 400,000 years ago, as part of the archaeocultural complex, a geographically limited group of hominids who lived in modern-day Israel. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 19, 2011, 2:46 a.m. Eastern Time. Quick note. Severe drought, other changes can cause permanent ecosystem disruption. Science Daily, October 13, 2011. An 8-year study has concluded that increasingly frequent and severe drought-dropping water tables and dried-up springs have pushed some aquatic desert ecosystems into a catastrophic regime, change from which many species will not recover. And good morning, this is Dr. Will, CBMD, PhD. This is Wednesday, October 19, 2011, 2:48 a.m. Eastern Time. Quick note. The findings just published in the Journal of Freshwater Biology raise concerns that climate change over pumping of aquifers for urban water use and land management may permanently affect which species can survive. And good morning, this is Dr. Will, CBMD, PhD. It's Wednesday, October 19, 2011, 2:53 a.m. Eastern Time. Quick note. Populations that have persisted for hundreds of thousands of years are now dying out. Springs that used to be permanent are now dying up. Streams that used to be perennial are now intermittent. And species that used to rise and fall in their populations are now disappearing. And good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 19, 2011, 3:08 a.m. Eastern Time. Quick note. The stream completely dried up for a period in 2005, and again in 2008 and 2009, leading to what researchers called a rapid regime shift in which some species went locally extinct and others took their place. The ecosystem dynamics are now different and show no sign of returning to their former state. 6 species were eliminated when the stream dried up, and 40 others became more abundant. Large-bodied top predators like the giant water bug disappeared and were replaced by smaller mesopredators such as aquatic beetles. Good morning, this is Dr. Will, CBMD, PhD. This Wednesday, October 19, 2011, 3:15 a.m. Eastern Time. Quick note. Conceptually similar events have taken place in the past in plant communities in Florida Everglades, floodplains in Australia, and boreal forest following fire disturbance. Other researchers have found in the Southwest climate change models predict longer, more frequent, and more intense droughts in the coming century, the scientists noted in their study. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 6, 2011, 6:12 a.m. Eastern Time. Quick note. Biologists find surprising number of unknown viruses in sewage. Science Daily, October 6, 2011. Though viruses are the most abundant life form on Earth, our knowledge of the viral universe is limited to a tiny fraction of the viruses that likely exist. In a paper published in the online journal of MBio, researchers from the University of Pittsburgh, Washington, University of St. Louis, University of Barcelona found that raw sewage is home to thousands of novel, undiscovered viruses, some of which could relate to human health. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 6, 2011, 6:14 a.m. Eastern Time. Quick note. There are roughly 1.8 million species of organisms on our planet, and each one is host to untold numbers of unique viruses, but only about 3,000 have been identified to date to explore this diversity and to better characterize the unknown viruses. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 6, 2011, 6:15 a.m. Eastern Time. Quick note. What was surprising was that the vast majority of viruses we found were viruses that had not been detected or described before. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 6, 2011, 6:16 a.m. Eastern Time. Quick note. The viruses that were already known include human pathogens like human papillomavirus and norovirus, which cause diarrhea. Also present were several viruses belonging to those familiar denizens of sewer everywhere. Rodents and cockroaches, bacteria are also present in sewage. So it was not surprising that the virus that prey on bacteria dominated the known genetic signatures. Finally, a large number of the known viruses found in raw sewage came from plants, probably owing to the fact that humans eat plants, and plant viruses outnumber other types of viruses than humans do. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 6, 2011, 6:18 a.m. Eastern Time. Quick note. The study was also the first attempt to look at the viruses in the population. Other studies have focused on bacteria or certain types of viruses. The researchers also developed new computational tools to analyze this data. The approach called metagenomics had been done before, but not with raw sewage. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 6, 2011, 6:19 a.m. Eastern Time. Quick note. The main application of this new technology, says Hendricks, will be to discover new viruses and to study gene exchange among viruses. The big question we are interested in is, where do emerging viruses come from? The team's hypothesis is that new viruses emerge, in large part through gene exchange. But before research on gene exchange can begin in earnest, large numbers of viruses must be studied, the researchers say. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 6, 2011, 6:20 a.m. Eastern Time. Quick note. First, you have to see the forest before you can pick out a particular tree to work on. If gene exchange is occurring among viruses, then we want to know where those genes are coming from. And if we only know about a small percentage of viruses that exist, then we are missing most of the forest. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 6, 2011, 6:22 a.m. Eastern Time. Quick note. First comet found with ocean-like water. Science Daily, October 5, 2011. New evidence supports the theory that comets delivered a significant portion of Earth's oceans, which scientists believe formed about 8 million years after the planet itself. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 6, 2011, 6:23 a.m. Eastern Time. Quick note. Burden and his co-investigator HiFi, the heterodyne instrument for the infrared and Herschel Space Observatory. With measurements from HiFi, the researchers found that the ice on the comet called Hartley 2 has the same chemical composition as our oceans. Both have similar DH ratios. The DH ratio is the proportion of deuterium or heavy hydrogen in the water. Deuterium atom is a hydrogen with an extra neutron in its nucleus. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 6, 2011, 6:26 a.m. Eastern Time. Quick note. The astronomers hypothesized that Hartley 2 was born in a different part of the solar system than the other six. Hartley most likely formed in the Kepler belt, which starts near Pluto at about 30 times farther than the Sun than Earth is. The other six hail from the Oort cloud, more than 5,000 times farther out. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 6, 2011, 6:28 a.m. Eastern Time. Quick note. The source of Earth's ocean has been a subject for debate among astronomers for decades. Until now, asteroids were thought to have provided most of the water. Now, however, Herschel has shown that at least one comet does have ocean-like water. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 6, 2011, 6:30 a.m. Eastern Time. Quick note. The results show that the amount of material out there that could have contributed to Earth's ocean is perhaps larger than we thought. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 6, 2011, 6:31 a.m. Eastern Time. Quick note. Herschel, European Space Agency mission with NASA participation, is an orbiting telescope that allows astronomers to observe at the far infrared wavelength where organic molecules and water emit their chemical signatures. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 6, 2011, 9:42 a.m. Eastern Time. Quick note. Triple rainbows exist, photo evidence shows. Science Daily, October 5, 2011. Few people have ever claimed to see 3 rainbows arcing through the sky at once. In fact, scientific reports of these phenomena, called tertiary rainbows, were so rare only 5 in 250 years. But until now, many scientists believe sightings were as fanciful as leprechauns' gold at a rainbow's end. These legendary optical rarities, caused by 3 reflections of each light ray within a raindrop, have finally been confirmed. thanks to the photographic perseverance, the new meteorological model that provides the scientific underpinnings to find them. And good morning, this is Dr. Will, CBMD, PhD. This is Thursday, October 6, 2011, 9:44 a.m. Eastern Time. Quick note. All staggering rare, tertiary, and quaternary rainbows are natural products of the combination of refraction, dispersion, and reflection inside raindrops. These are the same processes that create all rainbows, yet they are taken into their most extreme to produce these higher order variants. Refraction is when sunlight bends as it moves from air into water, and vice versa. Such bending makes ores look bent when partially submerged. Water droplets bend each of the colors in sunlight by a slightly different angle. This is called dispersion, and it separates the colors to create a rainbow. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 6, 2011, 9:46 a.m. Eastern Time. Quick note. Most of that multicolored light passes through the other side of the raindrop, but some is reflected. The raindrop's spherical curves concentrate those reflections at 138 degrees from the sun. The concentrated light is bright enough to create a visible primary rainbow. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 6, 2011, 9:47 a.m. Eastern Time. Quick note. A double rainbow occurs because not all that light exits the raindrop. Some is reflected back into the raindrop and goes through the whole process again. Although this light is dimmer, sometimes it is bright enough to produce a secondary rainbow just outside the first. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 6, 2011, 9:27 p.m. Eastern Time. Quick note. Patient-specific stem cells, major step toward cell-based therapies for life-threatening diseases. Science Daily, October 6, 2011. The team of scientists led by the Scott Nauger, the New York Stem Cell Foundation, NYSCF Laboratory in New York City, has made an important advance in the development of patient-specific stem cells that could impact the study and treatment of diseases such as diabetes, Parkinson's, and Alzheimer's. And good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 6, 2011, 9:26 p.m. Eastern Time. Quick note. The ultimate goal of the study is to save and enhance lives by finding better treatments and eventually cures for diabetes, Alzheimer's, Parkinson's, and other debilitating diseases and injuries affecting millions of people across the U.S. and the globe. The NYSCF CEO, Susan, the research brings us an important step closer to creating new healthy cells for patients to replace their cells that are damaged or lost through injury. And good morning, this is Dr. Will, CBMD, PhD. This Thursday, October 6, 2011, 9:31 p.m. Eastern Time. Quick note. The scientists demonstrate for the first time that the transfer of the nucleus from an adult skin cell of a patient into an oocyte without removing the oocyte nucleus results in reprogramming the adult nucleus to the pluripotent state. Embryonic stem cell lines were then derived from the oocyte containing the patient genetic material. Good morning, this is Dr. Will, CBMD, PhD. It's Thursday, October 6, 2011, 9:36 p.m. Eastern Time. Quick note. Monkeys move and feel virtual objects using only their brains. Science Daily, October 5, 2011. In the first ever demonstration of two-way interaction between a primate brain and virtual body, two monkeys trained in Duke University Center for Neuroengineering, learning to employ brain activity alone to move an avatar, hand and identity, the texture of virtual objects. And good evening, this is Dr. Will, CBMD, PhD. It's Thursday, October 6, 2011, 10:02 p.m. Eastern Time. Quick note. Astronomers find elusive planets in decade-old Hubble data. Science Daily, October 6, 2011. In a painstaking reanalysis of Hubble Space Telescope images from 1998, astronomers have found visual evidence for two extrasolar planets that went undetected back then. And good evening, this is Dr. Will, CBMD, PhD. It's Thursday, October 6, 2011, 10:03 p.m. Eastern Time. Quick note. Finding these hidden gems in the Hubble archive gives astronomers an invaluable time machine for comparing much earlier planet orbital motion data to more recent observations. It also demonstrates a novel approach for planet hunting in archival Hubble data. And good evening, this is Dr. Will, CBMD, PhD. This Thursday, October 6, 2011, 10:06 p.m. Eastern Time. Quick note. By finding the planets in multiple images spaced over years of time, the orbits of the planets can be tracked. Knowing the orbit is critical to understanding the behavior of multiple planet systems because massive planets can perturb each other's orbits. From the Hubble images, we can determine the shape of their orbits, which brings insight into the system, stability, planet masses, and eccentricities, and also the inclination of the system. And good evening, this is Dr. Will, CBMD, PhD. It's Thursday, October 6, 2011, 10:08 p.m. Eastern Time. Quick note. The three outer gas giant planets have approximately 100, 200, to 400-year orbits. This means that astronomers need to wait a very long time to see how the planets move along their paths. The added time span from the Hubble data helps enormously. The archive got us 10 years of science right now Without this data, we would have had to wait another decade. It is 10 years of science for free. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 1:19 a.m. Eastern Time. Quick note. Plants may have the genetic flexibility to respond to climate change. Science Daily, October 6, 2011. Plants may have the genetic flexibility to respond to climate change. In experiments with the common European plant Arabidopsis thaliana, a team of researchers led by Brown University scientists learned that climate is the agent that determines the suite of genes that gives the plant the best chance of surviving and reproducing throughout its natural range. The finding may unlock the molecular basis for other plants' adaptability to climate change. Results appear in Science. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 1:21 a.m. Eastern Time. Quick note. In a paper published in Science, the research team identifies the genetic signature in the common European plant Arabidopsis thaliana that governs the plant fitness, its ability to survive and reproduce. In different climates, the researchers further find that climate, in large measure, influences a suite of genes passed on to Arabidopsis to optimize its survival and reproduction. The set of genes determining fitness varies, the team reports, depending on the climate conditions of the plant's region, cold, warm, dry, wet, or otherwise. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 1:23 a.m. Eastern Time. Quick note. The researchers believe that by identifying genetic signatures that mark Arabidopsis' response to changing climate, scientists may understand how climate may cause the re-engineering of the genetic profiles of other plants. There is still evolutionary flexibility to help plants take one direction or another. Postdoctoral research at Brown's and paper's first author, it gives us good hope to see, yes, it's adapting. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 1:25 a.m. Eastern Time. Quick note. This was a truly massive undertaking, tracking more than 75,000 plants in the field from near the Arctic Circle to the Mediterranean coast. A former postdoctoral researcher lab on the faculty of Deep Spring College. Arabidopsis is an unusual annual plant, so we could measure total lifetime success of the individual within a single year. We gathered plants from a variety of native climates and grew some of each in our 4 widely distributed European garden sites. We shipped our harvested plants back to Brown and began the laborious task of counting fruits on these plants. In the end, we were able to assemble a very large and comprehensive data set that gives us new insight into what it takes for a plant to be successful in nature under a broad range of climate conditions. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 1:29 a.m. Eastern Time. Quick note. The team then burrowed into the Arabidopsis genome to find the molecular mechanism that might give the plant genetic flexibility to roll with climate punches. To identify variations in the genome among the regional representatives, the researchers carried out a genome-wide association study for survival and fruiting compromising more than 213,000 single nucleotide polymorphisms. These SNPs, frontier level explained, are like signposts pointing to areas in the genome where survival and reproduction may be emphasized and areas that show variations in the regional representatives' genetic makeup. And good morning, this is Dr. Will, CBMD, PhD. It's Friday, October 7, 2011, 1:30 a.m. Eastern Time. Quick note. In addition, certain climate variables seem to control the geographic distribution of fitness-associated SMP. For example, fitness SMP in Finland at the northern range limit by temperature. In one example presented in the paper, the researchers identify an SMP allele in the water stress tolerance gene called the SAG21. The allele was common in the Arabidopsis Spanish populations, but not in the cool climate of Finland, where tests showed plant carrying the allele fared poorly. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 1:32 a.m. Eastern Time. Quick note. We found that the genetic basis of survival and reproduction is almost entirely different in different regions, which suggests that evolutionary adaptation to one climate may not always result in a trade-off of poor performance in another climate. And good morning, this is Dr. Will, CBMD, PhD. It's Friday, October 7, 2011, 1:33 a.m. Eastern Time. Quick note. Thus, the Arabidopsis genome may contain evolutionary flexibility to respond to climate change. And good morning, this is Dr. Will, CBMD, PhD. It's Friday, October 7, 2011, 6:16 a.m. Eastern Time. Quick note. Is Shirari the norm for insects? Science Daily, October 7, 2011. The long-standing consensus of why insects stick together after mating has been turned on its head by scientists from the University of Exeter, published October 6 in Current Biology. Their study shows that, contrary to previous thinking, females benefit from this arrangement just as much as males. Instead of dominating their female, partners through bullying and aggressive behavior. Males were revealed to be protective, even laying their lives on the line when their mates face danger. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 6:18 a.m. Eastern Time. Quick note. Previous scientists assumed that male insects stay close to females after mating to stop them from taking other partners. Female insects have multiple mates, and the last mate is most likely to fertilize her eggs. Therefore, by preventing females from taking other mates, a male is most likely to father her offspring. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 6:19 a.m. Eastern Time. Quick note. To gain insight into the lives of wild field crickets, the research team used digital video technology, tagging, and DNA fingerprinting. They analyzed over 200,000 hours of infrared video footage taken over 2 entire breeding seasons to get a detailed picture of the daily dramas that occur in the insect world. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 6:25 a.m. Eastern Time. Quick note. Lungfish provides insight to life on land. Humans are just modified fish. Science Daily, October 4, 2011. A study into the muscle development of several different fish has given insights into the genetic leap that set the scene for the evolution of hind legs in terrestrial animals. This innovation gave rise to the tetrapods, 4-legged creatures, and our distant ancestors that made the first small steps on land some 400 million years ago. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 6:27 a.m. Eastern Time. Quick note. Scientists have long known that ancient lungfish species are the ancestors of the tetrapods. These fish could survive on land breathing air and using their pelvic fins to propel themselves. Australia is home to 3 species of the few remaining lungfish, 2 marine species and 1 inhabiting Queensland Mary River Basin. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 6:28 a.m. Eastern Time. Quick note. There are big gaps in our knowledge, however. Most conclusions have previously been drawn from fossil skeletons, but the muscle critical to locomotion cannot be preserved in the fossil record. The team used fish living today to trace the evolution of pelvic fin muscles to find out how the load-bearing hind limbs of the tetrapods evolved. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 6:34 a.m. Eastern Time. Quick note. We examine the way the different fish species generated the muscles of their pelvic fins, which are evolutionary forerunners of the hind limbs. A developmental biologist, his team genetically engineered the fish to trace the migration of precursor muscle cells in early developmental stages as the animal's body took shape. These cells in the engineered fish were made to emit a red or green light, allowing the team to track the development of specific muscle groups. They found that the bony fish had a different mechanism of pelvic fin muscle formation from that of the cartilaginous fish, a mechanism that was a stepping stone to the evolution of tetrapod physiology. And good morning, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 6:35 a.m. Eastern Time. Quick note. Humans are just modified fish. The genome of fish is not vastly different from our own. We have shown that the mechanism of pelvic muscle formation in bony fish is transitional between that in sharks and in our tetrapod ancestors. And good evening, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 10:46 p.m. Eastern Time. Quick note. Scientists have long agreed on a general picture on what causes pulsar emission. Electromagnetic forces created by the star's rapidly rotating magnetic field accelerate charged particles to near the speed of light, producing radiation over a broad spectrum, but the details remain a mystery. And good evening, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 10:52 p.m. Eastern Time. Quick note. Biofuel production left compared to fuel produced via artificial synthesis. Crops takes in CO2, water, and sunlight to create biomass, which then is transferred to a refinery to create fuel. In the artificial photosynthesis route, a solar collector or a windmill collects energy that powers an electrolyzer, which converts CO2 to a synthesis gas that is piped to a refinery to create fuel. And good evening, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 10:56 p.m. Eastern Time. Quick note. It lowers the overpotential for CO2 reduction tremendously. Who is also a professor of mechanical science and engineering and affiliated with the Beckman Institute for Advanced Science and Technology. Therefore, a much lower potential has to be applied. Applying a much lower potential corresponds to consuming less energy to drive the process. And good evening, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 10:59 p.m. Eastern Time. Quick note. Crab pulsar beams most energetic gamma rays ever detected from a pulsar. Science Daily, October 7, 2011. Astrophysicists have detected pulsed gamma ray emission from the Crab pulsar at energies far beyond what current theoretical models of pulsars can explain. And good evening, this is Dr. Will, CBMD, PhD. It's Friday, October 7, 2011, 11:01 p.m. Eastern Time. Quick note. Ancient supernovas discovered 10 billion-year-old exploding stars were a source of Earth's iron, researchers said. Science Daily, October 7, 2011. Supernova stars in the process of exploding open a window onto history of the elements of Earth's periodic table, as well as the history of the universe. All of those heavier than oxygen were formed in nuclear reactions that occurred during these explosions. And good evening, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 11:10 p.m. Eastern Time. Quick note. Natural compound helps reverse diabetes in mice. Science Daily, October 7, 2011. Researchers at the Washington University School of Medicine in St. Louis have restored normal blood sugar metabolism in diabetic mice using a compound the body makes naturally. The findings suggest that it may one day be possible for people to take the compound much like a daily vitamin as a way to treat or even prevent type 2 diabetes. And good evening, this is Dr. Will, CBMD, PhD. It's Friday, October 7, 2011, 11:13 p.m. Eastern Time. Quick note. All cells in the body make NMV, and a chain of reactions leading to production of NAD, a vital molecule that harvests energy from nutrients and puts it into a form cells can use. Among other things, NAD activates a protein called SIRT1 that has been shown to promote healthy metabolism throughout the body, from the pancreas to the liver to muscle and fat tissue. And good evening, this is Dr. Will, CBMD, PhD. This Friday, October 7, 2011, 11:19 p.m. Eastern Time. Quick note. Though the mice received NMV by injection in the study, the group is now conducting a long-term study of diabetic mice that the NMV dissolved in their drinking water. Calls this work as the first step toward a possible nutraceutical that people could take, almost like a vitamin, to treat or even prevent type 2 diabetes. And good morning, this is Dr. Will, CBMD, PhD. It's Saturday, October 8, 2011, 12:06 a.m. Eastern Time. Quick note. Astrophysicists and extinctions news about planet-threatening events. Science Daily, October 7, 2011. Space is a violent place. If a star explodes or black holes collide anywhere on our planet, or the Milky Way, they'd give off colossal blast of lethal gamma rays, X-rays and cosmic rays, and it's perfectly reasonable to expect Earth to be bathed in them. A new study of such events has yielded some new information about the potential effects of what are called short-hard interstellar radiation events. And good morning, this is Dr. Will, CBMD, PhD, Saturday, October 8, 2011, 12:11 a.m. Eastern Time. Quick note. Several studies in the past have demonstrated how longer high-energy radiation bursts, such as those caused by supernova and extreme solar flares, can deplete stratospheric ozone, allowing the most powerful and damaging forms of ultraviolet radiation to penetrate the Earth's surface. The probability of an event intense enough to disrupt life on the land or in the oceans becomes large, if considered on geological timescales, so getting a handle on the rates of intensities of such events is important for efforts to connect them to extinctions in the fossil record. And good morning, this is Dr. Will, CBMD, PhD. This is Saturday, October 8, 2011, 12:13 a.m. Eastern Time. Quick note. We find that the kind of gamma ray burst, a short gamma ray burst, is probably more significant than a longer gamma ray burst, said astrophysicist Brian Thomas of Washburn University, improved and accumulated data collected by the SWIFT satellite, which catches gamma rays burst in action in other galaxies, is providing a better case for the power and the threat of a short burst to life on Earth. And good morning, this is Dr. Will, CBMD, PhD. This Saturday, October 8, 2011, 12:15 a.m. Eastern Time. Quick note. The shorter bursts are really short, less than 1 second long. They are thought to be caused by the collision of two neutron stars, or maybe even colliding black holes. No one is certain which. What is clear is that they are incredibly powerful events. The duration is not as important as the amount of radiation. If such a burst were to happen inside the Milky Way, its effect would be much longer, lasting to Earth's surface and oceans. And good morning, this is Dr. Will, CBMD, PhD, Saturday, October 8, 2011, 12:18 a.m. Eastern Time. Quick note. What I focused on was the longer-term effects. The first effect is to deplete the ozone layer by knocking free oxygen and nitrogen atoms so they can recombine into ozone-destroying nitrous oxides. These long-lived molecules keep destroying ozone until they rain out, so we see a big impact on the ozone layer. And good morning, this is Dr. Will, CBMD, PhD, Saturday, October 8, 2011, 12:21 a.m. Eastern Time. Quick note. Based on what is seen among other galaxies, these short bursts, it seems that they occur in any given galaxy at a rate of about once per 100 million years. If that is correct, then it is very likely that Earth has been exposed to such events scores of times over its history. The question is whether they left a calling card in the sky or Earth's geological record. And Good morning, this is Dr. Will, CBMD, PhD, Saturday, October 8th, 2011, 12:23 a.m. Eastern Time. Quick note. Astronomical evidence is not likely, because the galaxy spins and mixes pretty thoroughly every million years, so any remnants of blast are probably long gone from view. There might, however, be evidence in the ground here on Earth. Some researchers are looking at the isotope iron-60, for instance, which has been argued as possible proxy for radiation events. And good morning, this is Dr. Will, CBMD, PhD, Saturday, October 8, 2011, 12:25 a.m. Eastern Time. Quick note. If isotopes like iron-60 can reveal the strata of the events, it then becomes a matter of looking for extinction events that correlate and seeing what died and what survived, which could shed more light on the event itself. And good evening, this is Dr. Will, CBMD, PhD, Saturday, October 8, 2011, 8:32 p.m. Eastern Time. Quick note. Reefs recovered faster after mass extinction than first thought. Science Daily, October 8, 2011. Metazoan-dominated reefs only took 1.5 million years to recover after the largest species extinction 252 million years ago. An international research team, including paleontologists from the University of Zurich, has established based on fossils from the southwestern United States. And good evening, this is Dr. Will, CBMD, PhD, Saturday, October 8, 2011, 8:33 p.m. Eastern Time. Quick note. Harsh living conditions caused by major fluctuations in the carbon content in sea levels, over-acidification, and oxygen deficiency in the seas triggered by largest mass extinction of all time at the end of the Permian era 252 million years ago. Life on Earth was also anything but easy after the obliteration of over 90% of all species. Throughout the entire early Triassic era, Metazoan-dominated reefs were replaced by microbial deposits. Researchers had always assumed it took Earth as long as 5 million years to recover from this species collapse. And good evening, this is Dr. Will, CBMD, PhD. This is Saturday, October 8, 2011, 8:35 p.m. Eastern Time. Quick note. Growth thanks to new reef-forming metazoan organisms. Metazoan-dominated reefs already developed during the early Triassic, much earlier than was previously assumed. As soon as the environmental conditions more or less returned to normal, the reef began to grow again due to metazoan organisms, and had played a secondary role in reefs up to then. This shows that after the extinction of dominated reef craters, metazoan were able to form reef ecosystems much sooner than was previously thought. And good evening, this is Dr. Will, CBMD, PhD, Saturday, October 8, 2011, 8:41 p.m. Eastern Time. Quick note. Electricity from the nose. Engineers make power from human respiration. Science Daily, October 8, 2011. The same piezoelectric effect that ignites your gas grill with the push of a button could one day power sensors in your body via the respiration in your nose. And good evening, this is Dr. Will, CBMD, PhD, Saturday, October 8, 2011, 8:42 p.m. Eastern Time. Quick note. In certain materials such as the polyviolidine fluoride PVDF used in Wang's team, electric charge accumulates in response to applied mechanical stress. This is known as the piezoelectric effect. Researchers engineered PVDF to generate sufficient electrical energy from respiration to operate small electronic devices. And good evening, this is Dr. Will, CBMD, PhD, Saturday, October 8, 2011, 8:45 p.m. Eastern Time. Quick note. Researchers are taking advantage of advances in nanotechnology and miniaturized electronics to develop a host of biomedical devices that could monitor blood glucose for diabetes or keep a pacemaker battery charged so that it would not need replacing. What's needed to run these tiny devices is a mini-scale power supply. Waste energy in the form of blood flow, motion, heat, or in the case of respiration, offers a consistent source of power. And good evening, this is Dr. Will, CBMD, PhD. This is Monday, October 10, 2011, 11:43 p.m. Eastern Time. Quick note. Laser light used to cool object to quantum ground state. Science Daily, October 9, 2011. For the first time, researchers at the California Institute of Technology, Caltech, in collaboration with a team from the University of Vienna, have managed to cool a miniature mechanical object to its lowest possible energy state using laser light. The achievement paves the way for the development of exquisitely sensitive detectors, as well as for quantum experiments that scientists have long dreamed of conducting. And good evening, this is Dr. Will, CBMD, PhD. It's Monday, October 10, 2011, 11:45 p.m. Eastern Time. Quick note. We've taken a solid mechanical system, one made up of billions of atoms, and used optical light to put it into a state in which it behaves according to the laws of quantum mechanics. In the past, this has only been achieved with trapped single atoms or ions. And Good evening, this is Dr. Will, CBMD, PhD. This is Monday, October 10th, 2011, 11:47 p.m. Eastern Time. Quick note. As described in the paper, a painter and his colleagues have engineered a nanoscale object, a tiny mechanical silicone beam, such that laser light of carefully selected frequency can enter the system and, once reflected, can carry thermal energy away, cooling the system. And good evening, this is Dr. Will, CBMD, PhD. This is Monday, October 10th, 2011, 11:49 p.m. Eastern Time. Quick note. By carefully designing each element of the beam as well as a patterned silicone shield that isolates it from the environment, painter and colleagues were able to use the laser cooling technique to bring the system down to the quantum ground state, where mechanical vibrations are at the absolute minimum. Such a cold mechanical object could help detect very small forces or masses, whose presence would normally be masked by the noisy thermal vibrations of the sensor. And good evening, this is Dr. Will, CBMD, PhD. This Monday, October 10, 2011, 11:50 p.m. Eastern Time. Quick note. In many ways, the experiment we've done provides a startling point for the really interesting quantum mechanical experiments one wants to do. For example, scientists would like to show that a mechanical system could be coaxed into a quantum superposition, a bizarre quantum state in which a physical system can exist in more than one position at once. But they need a system at the quantum ground state to begin such experiments. And good evening, this is Dr. Will, CBMD, PhD. This is Monday, October 10, 2011, 11:52 p.m. Eastern Time. Quick note. To reach the ground state, Painter's group had to cool its mechanical beam to a temperature below 100 millikelvin minus 273.15 degrees Celsius. That's because the beam is designed to vibrate at gigahertz frequencies, corresponding to 1 billion cycles per second, a range where a large number of phonons are present at room temperature. Phonons are the most basic units of vibration, just as the most basic units of packets or light are called photons. All of the photons in the system have to be removed to cool it to the ground state. And good evening, this is Dr. Will, CBMD, PhD. This is Monday, October 10, 2011, 11:55 p.m. Eastern Time. Quick note. Conventional means of cryogenetically cooling to such temperatures exist but require expensive and in some cases impractical equipment. There is also the problem of figuring out how to measure such a cool mechanical system. To solve both problems, the Caltech team used a different cooling strategy. And good evening, this is Dr. Will, CBMD, PhD. This is Monday, October 10, 2011, 11:56 p.m. Eastern Time. Quick note. What we've done is use the photons, the light field, to extract phonons from the system. To do so, the researchers drilled tiny holes at precise locations in their mechanical beam so that when they directed laser light of a particular frequency down the length of the beam, the holes acted as mirrors, trapping the light in the cavity and causing it to interact strongly with the mechanical vibrations of the beam. And good evening, this is Dr. Will, CBMD, PhD. This is Monday, October 10, 2011, 11:58 p.m. Eastern Time. Quick note. Because the shift in the frequency of light is directly related to the thermal motion of the mechanical object, the light, when it eventually escapes from the cavity, also carries with it information about the mechanical system, such as the motion and temperature of the beam. Thus, the researchers have created an efficient optical interference to a mechanical system element or an optomechanical transducer that can convert information from the mechanical system into photons of light. And good morning, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 12:02 a.m. Eastern Time. Quick note. The other cooling techniques use a startling temperatures of approximately 20 millikelvin, more than a factor of 10,000 times cooler than room temperature. Ideally, to simplify, design scientists would like to initiate these experiments at room temperature. Using laser cooling, Painter and his colleagues were able to perform their experiment at a much higher temperature, only about 10 times lower than room temperature. And good evening, this is Dr. Will, CBMD, PhD. This Tuesday, October 11, 2011, 12:12 a.m. Eastern Time. Quick note. Venus has an ozone layer to space probe discoveries. Science Daily, October 9, 2011. The European Space Agency Venus Express spacecraft has discovered an ozone layer high in the atmosphere of Venus, comparing its properties with those of equivalent layers on Earth and Mars. Will help astronomers refine their searches for life on other planets. And good morning, this is Dr. Will, CBMD, PhD. This Tuesday, October 11, 2011, 12:17 a.m. Eastern Time. Quick note. The small amount of ozone in Mars' atmosphere has not been generated by life. There is the result of sunlight breaking up carbon dioxide molecules. Venus, too, now supports this view of a modest ozone buildup by non-biological means. Its ozone layer sits at the altitude of 100 kilometers, about 4 times higher in the atmosphere than Earth's, and is 100 to 1,000 times less dense. And good morning, this is Dr. Will, CBMD, PhD. This Tuesday, October 11, 2011, 12:18 a.m. Eastern Time. Quick note. Theoretical work by astrobiologists suggests that planet's ozone concentration must be 20% of Earth's value before this should be considered as a cause. These new results support the conclusion because Venus clearly remains below this threshold. And good morning, this is Dr. Will, CBMD, PhD. This Tuesday, October 11, 2011, 12:26 a.m. Eastern Time. Quick note. Babies show sense of fairness, altrusion as early as 15 months. Science Daily, October 10, 2011. A new study presents the first evidence that a basic sense of fairness and altrusion appears in infancy. Babies as young as 15 months perceive the difference between equal and unequal distribution of food, and their awareness of equal rations was linked to their willingness to share a toy. And good morning, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 12:36 a.m. Eastern Time. Quick note. Supersaturated water vapor in Martian atmosphere. Science Daily, October 10, 2011. Analysis of data collected by the European Space Agency of Mars Express spacecraft leaves no room for doubt. The Martian atmosphere contains water vapor in a supersaturated state. This surprising finding will enable scientists to better understand the water cycle on Mars, as well as the evolution of its atmosphere. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 11, 2011, 12:52 a.m. Eastern Time. Quick note. Sniffing out the brain's predictive power, human brain smells what it expects rather than what it sniffs. Science Daily, October 10, 2011. In the moments before you stop and smell the roses, it is likely your brain is already preparing your sensory system for that familiar flora smell. New research from the Northwestern Medicine offers strong evidence that the brain uses predictive coding to generate predictive templates of specific smells, setting up a mental expectation of a scent before it hits your nostrils. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 11, 2011, 1:20 a.m. Eastern Time. Quick note. Series of bumps sent Uranus into the sideways spin, new research suggests. Science Daily, October 10, 2011. Uranus' highly tilted axis makes it something of an oddball in our solar system. The accepted wisdom is that Uranus was knocked on its side by a single large impact, but new research being presented at the EPSC-DPS joint meeting in Nantes rewrites our theories of how Uranus became so tilted and also solves fresh mysteries about the position and orbits of its moons. By using simulations of planetary formation and collisions, it appears that early in its life, Uranus experienced a succession of small punches instead of a single knockout blow. This research has important ramifications on our theories of a giant planet formation. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 11, 2011, 1:45 a.m. Eastern Time. Quick note. Giant Kraken layer discovered cunning sea monster that preyed on Ichthyosaurus. Science Daily, October 10, 2011. Long before whales, the oceans of Earth were roamed by a very different kind of air-breathing leviathan. Snaggle-toothed Ichthyosaurus, larger than school buses, swam at the top of the Triassic period ocean food chain, or so it seemed before Mount Holyoke College paleontologist Mark McMenamin took a look at some of their remains in Nevada. Now he thinks there was an even larger and more cunning sea monster that preyed on Ichthyosaurus, a kraken of such mythological proportions it would have sent Captain Nemo running for dry land. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 11, 2011, 6:18 a.m. Eastern Time. Quick note. Long Lost Lake offers clues to climate change. Science Daily, October 5, 2011. What caused water levels to drop in an immense, yet long-vanished lake? Research by University of Cincinnati geologists suggest the conditions 12,000 years ago encourage evaporation. And good morning, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 6:20 a.m. Eastern Time. Quick note. Not long ago, geologically speaking, a now-vanished lake covered a huge expanse of today's Canadian prairie. As big as Hudson Bay, the lake was fed by melting glaciers as they receded at the end of the last ice age. At its largest glacial lake, as it is known, covered most of the Canadian province of Manitoba, plus a good part of western Ontario. A southern arm straddled the Minnesota-North Dakota border. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 11, 2011, 6:24 a.m. Eastern Time. Quick note. The dry air would reduce rainfall and enhance evaporation. The cold would reduce meltwater production, and short-wave radiation would enhance evaporation when the lake was not frozen, and sublimation when the lake was ice-covered. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 11, 2011, 6:24 a.m. Eastern Time. Quick note. Further research will attempt to clear a picture of this ancient episode, but researchers will have to incorporate various factors including humidity, yearly duration of lake ice, annual temperature, and better understanding of how and where meltwater flowed from the receding glaciers. And good morning, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 6:26 a.m. Eastern Time. Quick note. We look at the mineralogy of the sediments, and also the chironomids. They are a type of midge, and they are very temperature sensitive. The exact species of the abundance of midges in our cores can help pinpoint temperature when these sediments were deposited. And good morning, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 6:28 a.m. Eastern Time. Quick note. How the brain makes memories rhythmically. Science Daily, October 11, 2011. The brain learns through changes in the strength of its synapses, the connections between neurons, in response to stimuli. Now in a discovery that challenges conventional wisdom on the brain, mechanisms of learning. UCLA neurophysicists have found there is an optimal brain rhythm or frequency for changing synaptic strength, and further like stations on the radio dial. Each synapse is turned to a different optimal frequency for learning. And good morning, this is Dr. Will, CBMD, PhD. This Tuesday, October 11, 2011, 6:30 a.m. Eastern Time. Quick note. A change in the strength of synapse in response to stimuli, known as synaptic plasticity, is induced through a so-called spike train, series of neural signals that occur with varying frequency and timing. Previous experiments demonstrated that stimulating neurons at a very high frequency, 100 spikes per second, strengthen the connecting synapse, while low-frequency stimulation, 1 spike per second, reduced synaptic strength. And good morning, this is Dr. Will, CBMD, PhD. This Tuesday, October 11, 2011, 6:32 a.m. Eastern Time. Quick note. These earlier experiments used hundreds of consecutive spikes in very high frequency range to induce plasticity. Yet, when the brain is activated during real-life behavioral tasks, neurons fire only about 10 consecutive spikes, not several hundred, and they do so at a much lower frequency, typically in the 50 spike per second range. And good morning, this is Dr. Will, CBMD, PhD. This Tuesday, October 11, 2011, 6:33 a.m. Eastern Time. Quick note. In other words, spike frequency refers to how fast the spikes come. 10 spikes could be delivered at a frequency of 100 spikes a second, or at a frequency of 1 spike per second. And good morning, this is Dr. Will, CBMD, PhD. This Tuesday, October 11, 2011, 6:35 a.m. Eastern Time. Quick note. The knowledge of the synapse has a preferred frequency for maximal learning led the researchers to compare optimal frequencies based on the location of the synapse on a neuron. Neurons are shaped like trees, with the nucleus being the base of the tree, the dendrites resembling the extensive branches, and the synapses resembling the leaves on those branches. And good morning, this is Dr. Will, CBMD, PhD. This Tuesday, October 11, 2011, 6:36 a.m. Eastern Time. Quick note. The expectation based on previous studies was that if you drove the synapse at a higher frequency, the effect on synaptic strengthening or learning would be at least as good as, if not better than, the naturally occurring lower frequency. To our surprise, we found that beyond the optimal frequency, synaptic strengthening actually declined as the frequencies got higher. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 11, 2011, 6:37 a.m. Eastern Time. Quick note. Compared synaptic learning based on where synapses were located on the dendritic branches. What they found was significant. The optimal frequency for inducing synaptic learning changed depending on where the synapse was located. The farther the synapse was from the neuron's cell body, the higher its optimal frequency. And good morning, this is Dr. Will, CBMD, PhD. This Tuesday, October 11, 2011, 6:38 a.m. Eastern Time. Quick note. Incredibly, when it comes to learning, the neuron behaves like a giant antenna, with different branches of dendrites tuned to different frequencies for maximal learning. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 11, 2011, 6:39 a.m. Eastern Time. Quick note. The researchers found that not only does each synapse have a preferred frequency for achieving optimal learning, but for the best effect, the frequency needs to be preferably rhythmic timed at each interval. Even at the optimal frequency, if the rhythm was thrown off, synaptic learning was substantially diminished. And good morning, this is Dr. Will, CBMD, PhD. It's Tuesday, October 11, 2011, 6:40 a.m. Eastern Time. Quick note. The research also showed that once a synapse learns, its optimal frequency changes. In other words, if the optimal frequency for a naive synapse, one that has not learned anything yet, was say 30 spikes per second after learning, that very same synapse would learn optimally at a lower frequency, say 24 spikes per second. Thus, learning itself changes the optimal frequency for a synapse. And good evening, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 8:14 p.m. Eastern Time. Quick note. Measuring elusive solar neutrinos flowing through the Earth. Physicists learn more about the Sun. Science Daily, October 7, 2011. Using one of the most sensitive neutrino detectors on the planet, physicists at the university are measuring the flow of solar neutrinos reaching Earth more precisely than ever before. The detector probes matter at the most fundamental level and provides a powerful tool for directly observing the Sun's composition. And good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 11, 2011, 8:16 p.m. Eastern Time. Quick note. Now measured with high precision the flux of beryllium-7, solar neutrino abundant low-energy particles only below the observable threshold. With this advance, they can now precisely study behavior of solar neutrinos with kinetic energy below 1 mega electron volt, MeV. Vora Exino. Scientists also recently reported the first observation of neutrinos produced in a little-studied solar reactor process known as the proton-electron proton, or PEP, and set of stringent limits on reactions involving carbon, nitrogen, and oxygen, the CNO cycle in the sun, a nuclear process. And good evening, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 8:27 p.m. Eastern Time. Quick note. Sexual selection by sugar molecule helped determine human origins, researchers say. Science Daily, October 11, 2011. Researchers at the University of California, San Diego School of Medicine say that losing the ability to make a particular kind of sugar molecule boosted disease, protection in early hominids, and may have directed the evolutionary emergence of our ancestors, the genus Homo. And good evening, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 8:29 p.m. Eastern Time. Quick note. The findings published in this week's early online edition of the Proceedings of the National Academy of Sciences are among the first evidence of a novel link between cell surface sugars, Darwinian sexual selection, and immune function in the context of human origin. And good evening, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 8:34 p.m. Eastern Time. Quick note. Sialic acids are sugar molecules found on the surfaces of all animal cells, where they serve as vital contact points for interaction with other cells and with the surrounding environment, including as targets for invasive pathogens. For millions of years, the common ancestors of humans and other apes shared a particular kind of sialic acid known as N-glycolic neuramic acid, or NEU5GC, then for reasons possibly linked to malarial parasite that bound NEU5GC, a gene mutant 3 million or so years ago inactivated the human enzyme involved in making the molecule, and instead of humans began producing more slightly different form of sialic acid called NEU5AC, the precursor of NEU5GC. And good evening, this is Dr. Will, CBMD, PhD. It's Tuesday, October 11, 2011, 8:53 p.m. Eastern Time. Quick note. New diamond, new form of super hard carbon, is as strong as a diamond. Science Daily, October 11, 2011. Carbon is the fourth most abundant element in the universe and takes a wide variety of forms called allotropes, including diamond and graphite. Scientists at the Geophysical Laboratory are part of a team that has developed a new form of carbon, which is capable of withstanding extreme pressure stresses that were previously observed only in diamond. And good evening, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 8:54 p.m. Eastern Time. Quick note. This new carbon form was capable of withstanding 1.3 million times normal atmospheric pressure in one direction, while confined under a pressure of 600,000 times atmospheric levels in other directions. No substance other than diamond has been observed to withstand this type of pressure stress, indicating that the new carbon allotrope must indeed be very strong. And good evening, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 8:57 p.m. Eastern Time. Quick note. However, unlike diamond and other crystalline forms of carbon, the structure of this new material is not organized in repeating atomic units. It is an amorphous material, meaning that its structure lacks a long-range order of crystals. This amorphous super-hard carbon allotrope would have a potential advantage over diamond if its hardness turns out to be allotrophic, that is, having hardness that is equally strong in all directions. In contrast, diamond hardness is highly dependent upon the direction in which the crystal is orientated. And good evening, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 9:03 p.m. Eastern Time. Quick note. Most vertebrates, including humans, descend from ancestor with sixth sense. Science Daily, October 11, 2011. People experience the world through five senses, but sharks, paddlefishes, and certain other aquatic vertebrates have a sixth sense. They can detect weak electrical fields in the water and use this information to detect prey, communicate, and orientate themselves. And good evening, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 9:10 p.m. Eastern Time. Quick note. Astronomers find bounty of failed stars. One youngster only 6 times heftier than Jupiter. Science Daily, October 11, 2011. University of Toronto-led team of astronomers has discovered other 24 new free-floating brown dwarfs, including a lightweight youngster only about 6 times heftier than Jupiter, that reside in 2 young star clusters. What's more, one cluster contains a surprising surplus of them, harboring half as many of these astronomical oddballs as normal stars. And Good evening, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 9:13 p.m. Eastern Time. Quick note. Our findings suggest once again that objects not bigger than Jupiter could form the same way as stars do. In other words, nature appears to have more than one trick up its sleeve for producing planetary mass objects. End. Good evening, this is Dr. Will, CBMD, PhD. This is Tuesday, October 11, 2011, 9:15 p.m. Eastern Time. Quick note. Brown dwarfs straddle the boundary between stars and planets, sometimes described as failed stars. They glow brightly when young from the heat of formation but cool down over time and end up with atmospheres that exhibit planet-like characteristics. Scientists think that most brown dwarfs may have formed like stars, in isolation from contracting gas clouds, but some of the punniest free floaters may have formed like planets around the star and later ejected, and